Welcome to Cracking Cancer, where we tackle the tough questions about cancer with real patients and world experts. Our mission is to empower the cancer community through education, bring transparency to the oncology system, and drive funding for critical cancer research. Whether you're facing a diagnosis, supporting a loved one, or simply here to learn, we're here to bring clarity, hope, and action to the fight against cancer.
Hey everyone, welcome to Cracking Cancer. I am your host, Dr. Kyle Concannon. I am a physician scientist at the University of Colorado, and joining me today is Samantha Murrell.
Hey everyone, I'm Samantha Murrell. I'm a patient advocate and I happen to have stage 4 lung cancer with a HER2 mutation. I'm happy to be here.
Thanks for joining again, and very topical because today we're going to be talking about what is a tyrosine kinase inhibitor, which is a medicine that you just transitioned to after being on chemotherapy, then you were on an antibody drug conjugate, and the tyrosine kinase inhibitors are a class of medication that are we're, as a community, really proud of. There's been a lot of research that has generated these new medicines, and people have really had good responses and low side effects, which is the crowning jewel, I guess, of what research is supposed to produce. It's not all positive. It's not all, not everyone has long-term responses and not everyone has no side effects. But generally speaking, this was a class of medication that came after chemotherapy, and it's a lot more elegant than chemotherapy. So we're going to be going into that kind of detail. But before we get into the nitty-gritty stuff, I'm curious, Samantha, when you were first diagnosed, we mentioned you were on chemo, and you knew of tyrosine kinase inhibitors. Mm-hmm. What was your perception of this class of medication that we're about to talk about?
I very much saw it as a magical pill that could extend my life, that would not be as toxic as other things on the market. So I was very excited about the potential of that. I knew that they were very targeted. They went after the specific mutation or fusion that one had, which was also a plus. And they really intrigued me. I was almost like, is this too good to be true?
Yeah. And I think it's probably a good way to think about it, if it's a potential for someone, is one, they're usually pills, which is a huge deviation from chemotherapy, where you're going in for chemo, you're getting labs all the time. So the idea that you can just take this thing at your house is a huge improvement over, I would say, most chemos. And they are very targeted, and that is beneficial not only because it probably works better against most cancers, but also the side effects are usually not side effects from attacking the tumor too well. It's side effects from when the therapy we give attacks other stuff. So when we say targeted, that's what we mean. We mean it targets the tumor, and it tends to not target healthy tissue, non-tumor tissue. So I think your perception is right. Obviously, when something sounds too good to be true, it is. Yeah. Targeted therapies, generally speaking, they're wonder drugs compared to chemo, but they're not cure-alls. Mm-hmm. So I'm eager to go into more of maybe the truth about the tyrosine kinase inhibitor class, TKIs, you'll hear us call them. But first, we should probably lay out some groundwork on, we're using these terms, targeted kinase inhibitors, and these are scientific terms, So I want to maybe first talk about what that means. And I think earlier, we had different podcasts about what is cancer and what causes cancer. And the targeted kinase inhibitors are interesting because it does get to one of the root causes of cancer. It does target specifically what's causing cancer. So I think it makes sense to lay that groundwork again. Do you think that'd be helpful?
Absolutely. Because honestly, when I try to explain to people what I'm taking, I just say a chemo pill, which I know is not a good description of it at all. So if you could break it down, maybe people would have a better understanding and be able to explain to their friends and family a little bit more what they're really taking, because I know it's very much not a chemo pill.
Yeah, it hurts a little bit to hear that, but we're very proud of the tyrosine kinase inhibitors because they're not chemo. I mean, that being said, they're not perfect either. So I think, we can lay out some groundwork here, And the first thing, we should probably get into a little bit of the biology of what causes cancer, because this does get to that root cause. We mention mutations in cancer a lot. And we say that mutations can cause cancer. It's not the only thing that causes cancer, at least targetable mutations like those that tyrosine kinase inhibitors target. But that's usually what we're talking about in this realm when we're talking about TKIs are mutations. And the reason that's important is mutations are in DNA. And your DNA, we have a lot of perceptions of what DNA is and what DNA isn't, but really I think of it as a blueprint. The DNA is the blueprint that the cells in your body use to make certain proteins when they choose to. Every cell in your body, generally speaking, has the same DNA, but they're different. Your eyes look different than your skin because they're choosing to use different sections of that blueprint. So that's the first step, is what DNA is functioning as is really just a blueprint. So part of that blueprint can be mutated. And that's what we mean is mutations, is when you have a normal blueprint, but part of that is different in that cell than it's supposed to be. And your body makes tons of mutations. This isn't a rare thing in the landscape of your body. If we do complicated mutational analysis in your blood, we'll find a ton of mutations. Most of them don't actually change what will happen, though. So it's actually fairly rare to have a mutation in the body that changes what is ultimately going to be made in the cell. So as you have your DNA is a blueprint for RNA. RNA is a blueprint for protein. So all of this is a pathway to get protein. What happens when you have a mutation is that blueprint is a little bit different, not usually a lot different, just a little different. And then it makes the RNA that's a little bit different, which makes a protein that's a little bit different. And when we have a mutation, that small difference usually has a really significant impact. How did that sound? Does that make sense, Samantha?
Yeah, it does make sense.
Yeah, and when we're talking about cancer, that impact means that mutation is telling the cell to divide. So we have mutations in these proteins called kinases. Since we're talking about targeted kinase inhibitors, the kinases are the things that we're really talking about here. That's a class of protein that has a job in the cell to signal something. It's telling the cell to change in some way, to adapt in some way. And they function in your cells all the time. I mean, these are normal things that are supposed to be there. But like everything else in your body, there's a really strong regulatory process that tells things to signal at certain times and not to signal at other times. A lot of times when these kinases are mutated, they tell the cell to divide. They tell it to grow. And the regulation doesn't work anymore. So a lot of times there's just one single area in a very, very large protein, hundreds to thousands of amino acids or sections of that protein, building blocks. Just one of them has to be mutated in the perfect place for that signal to get dysregulated. So for you as an example, you have a HER2 mutation. HER2 is a protein. It is a kinase. It tells the cell to signal, or it signals to the cell to grow, rather. And that's normal. Your cells need to divide sometimes. But when you have a specific small area of that protein that's mutated, which you do, you have a small area of the protein that has an abnormality in it, and all of a sudden it's telling the cell to divide all the time. And that's usually what we're trying to target. Mm-hmm. And a protein it's a three-dimensional structure. If you enlarged it, it would be, it would have different grooves, it would have different pockets that other areas or other proteins are interacting with it. So those different areas are doing different things. Specifically, when areas in these kinases are mutated, they tend to be mutated at locations where they're signaling. And since that's their primary function, that ends up causing them to signal too much. That too much signaling leads to additional cell growth, and it can't turn it off. It's just always saying signal as long as that mutation's there. And since that mutation was in the DNA, which is the blueprint for how you made it, you're mass producing these proteins in the cell that are all dysfunctional. All of them are saying signal all the time.
And all I want to say to that is rude.
It's very rude. Well, and a lot of times when we're talking about these types of mutations, which I want to highlight again, it's not the only way cancer happens. And in the whole bucket of things, of causes of cancer, this is actually not one of the more common ones. Less than half of cancers have these by quite a large margin. But when they do happen, since that mutation is so specific, sometimes it can be targeted through medicines. And that was the whole goal of this class of medication that emerged, less than 30 years ago, really, at least in everyday use. Samantha, what did I leave out there?
No, that was very informative. Made sense. I mean, my initial reaction is just specifically about mutations, I think, is, and this is kind of a sidebar, but I think people are curious about this, is why we don't know why people get certain mutations, right? We don't know necessarily what they've been exposed to, but it's always interesting to me to look at all the people around me and see XYZ person has ALK, XYZ person has EGFR, someone else has RET, I have HER2. Like, could we all have been exposed to the same things and we just happen to get different mutations? Like, how does our body decide to mess up in that particular way?
Well, it's interesting, and for those listening, those are all different types of kinases that Samantha just mentioned. So for many who are probably listening, you're listening to this podcast because you have a tumor, you have a loved one who has a tumor that's driven by one of these things. And the reality of it right now is we don't really know what causes them. Mm-hmm. A lot of times people think you have a lung cancer. Many people think lung cancer and smoking. Smoking does cause mutations. It doesn't tend to cause these. Mm-hmm. These are mutations that just happen to happen really once, rather than a lung cancer associated mutational burden, I'll say, where you get a lot of mutations over time. And then in lung cancers that are smoking related, you have the balance of proteins that are telling the cell to divide and proteins that are telling the cell to not divide, to stay as it is. So there's a certain regulatory balance. With smoking, that balance comes off step by step with different proteins getting mutated the mutations are almost at random. So you need a lot of them in order to have that full effect. Whereas if you have a kinase mutated at just the right spot, that is completely out of whack immediately. Like, that is the only thing you really need. If someone's a scientist and listening to this, you may gripe with some caveats here, but by and large, that's the only thing you need for that cell to become cancerous. And since it doesn't have a lot of mutations in it, it can just need that one. It actually looks pretty similar to the rest of your cells, all things considered. It's not like the lung cancer that was associated with smoking, where you had a ton of mutations, and that's very different from what your cells normally are, and that has implications for the immune system too. Mm-hmm. So these tyrosine kinase inhibitors tend to be really, really good for individuals who have those specific mutations that caused the cancer. They don't tend to be smoking associated, and then the immunotherapies tend to not work as well on those because those cells look really similar. They just have one difference rather than hundreds or thousands or millions, which can be with carcinogens. So it's really fortunate we have this class of tyrosine kinase inhibitor because this was the first, in my mind, it was really the first scientifically driven strategy to attack a root cause of cancer. People think that chemotherapies were really originally designed to be anti-cancer drugs, but a lot of them originated from something else. Some of our first chemotherapies were from nitrogen mustards. So these were wartime weapons. And then we found out through laboratory studies that they kill tumors because they knew how these things work. They worked on DNA, and DNA is more accessible if a cell is dividing. So while chemotherapies are somewhat specific for cancer because cancer is dividing a lot, they attack whatever divides a lot. And that leads to the toxicities of the side effects, really, of traditional chemotherapies. But yeah, it's funny. I remember one of the first things that got me interested in learning about oncology was back when I was an undergraduate. And I really thought I was a biology major. I loved the cell cycle. I loved learning how all that's regulated. And then when problems happen, what can you do? And I remember learning about these tyrosine kinase inhibitors, these TKIs, because where we had chemotherapy at that time, and the TKI class has really just come out back when I was an undergrad. And someone had come out with one that specifically targeted the root cause of how a cancer happened. It wasn't just dumping chemotherapy on everything. And I thought that was the most, just the proof of principle that you can learn more about a tumor, figure out what's causing it, and attack that root cause was pretty new to me. And it was a demonstration of how I can apply my scientific interests into actually helping someone tangibly. Like, this wasn't, I want to better understand how these proteins interact, which a lot of undergrad is intellectual curiosity. It's not applied science, at least when you're learning about it at that level. So to be able to see that applied science in action in really real time, because patients were just starting to get these things, really made me passionate that not only could I continue to learn more cell biology, but as I got better at it, I could weaponize that knowledge against cancer. And seeing someone actually do that gave me hope as a future oncologist that we could make a difference. Because this is something that, I consider to be molecular snipers rather than attacking absolutely everything else. Chemotherapy is like carpet bombing. You're attacking everything. But having a targeted therapy that really just attacks the root cause, to me, sounded amazing. And it really inspired me to study that more in undergraduate. And then my first job out of undergraduate was actually in a laboratory that worked on tyrosine kinase inhibitors. EGFR was the first one I worked on, the epidermal growth factor receptor, which is very similar to the mutation you have. And we worked on making better TKIs and understanding resistance in that type of cancer.
So I have a curiosity question, but I think it would be helpful for people who are just maybe learning about TKIs. Maybe they're just starting one. When you get that report back from your doctor that kind of says you have X mutation, sometimes there are other co-mutations on that list as well. And I think it makes people nervous of, okay, I'm starting this TKI for one of my mutations. What about the other ones? If you could go into a little bit in terms of like how that all works, how does it still shut down the cancer, et cetera, et cetera?
Yeah, no, it's a great question because I'm mentioning specifically targeted kinase inhibitors, and the kinases are the type of protein that we can attack. But in any given mutational report, you're going to have most people will have many mutations. And it depends on how comprehensive they looked. You have 23,000 genes. Many of those have mutations in them just because, random chance. And there are a lot of genes that are associated with cancers that we tend to check more now. We don't check every mutation that you could possibly have in the body. We have limited panels of things that may have relevance to cancer. And that's what comes out in the report. The report doesn't have every gene that you have in the body. So typically you have driver oncogenes is what we're talking about here. And what I mean when I say that, the term oncogene is a cancer-causing gene. So when it's mutated, it causes cancer. Onco is cancer. So you have cancer-causing genes that can be drivers, meaning they themselves, being mutated, are good enough to cause the cancer. So, for example, if someone has a HER2 mutation, like your tumor has, that is the driver oncogene. Then you have other things that can potentially contribute, I'll say, maybe not, but potentially contribute to the cancer's dysregulation. I mentioned before, if someone has carcinogen exposure, you can get a lot of mutations that will continue to tip that balance more and more of should the cell divide and should the cell not. When you have a driver oncogene, the cell will divide, but cells that will develop additional mutations will probably divide faster. And that's a selective advantage. Cancer is evolution in real time in the body. Something that makes those cells divide faster or be resistant to therapies or just survive in some way better than the other ones are going to be selected for, and those are the ones that are going to expand faster. So, and that's my best understanding of why these mutations are seen. The reality is I don't have a perfect answer for exactly why, but that's my best scientific knowledge around it. So usually people will have the one driver oncogene, but you may have those other things. And those other things may facilitate the cancer growing faster, and that's why they happened. That's why they were selected for, I guess. But usually, if you have a driver oncogene there, the cell depends on that signal to divide.
Oh, interesting.
So it will depend on that. And that was the big breakthrough with these tyrosine kinase inhibitors, the TKIs, is a dependency on the oncogene. So if you have a common co-mutation is P53. Many people will have P53 mutations. That in itself is not enough to cause the cancer. In my mind, that may lead to resistance faster. It may lead to faster growth of a cancer that's already there. But without the signaling of HER2 to say divide, it doesn't have a divide signal. So, and they become addicted to it, which is a really important concept. Many of the oncogene-driven lung cancers, or cancers wherever, are addicted to the oncogenic signaling, which was the brilliance of this TKI strategy. If you can block that one thing, then the one signal that's coming from the mutated protein, then the cells die because they were addicted to it. And I'll say 95 to 99% of the cells die, usually somewhere around there.
So you say they get addicted to it, and I'm jumping way ahead here, but I think it's very timely because of you saying that. Why then does resistance occur?
Oof. Yeah, billion-dollar question. Tyrosine kinase inhibitor resistance can occur in a lot of different ways. And I'll mention some of the ways we know about, but the reality is cancer, like I mentioned, is evolution at work. So if you have, most people, if they have a tumor that's visible, are having billions of tumor cells. So a lot of tumor cells. And then if you give something that's highly effective and it kills 99%, that's very good. But 1% of a billion is still a lot of tumor cells. And those survived from one of many possible pathways. So I want to start by just understanding the vast landscape of what's left over after you give after you give any type of cancer therapy. If you didn't eradicate everything, there's still usually many tumor cells present. Now, they may be inhibited and not dividing, and that's one way of causing resistance, is they just don't, they stop dividing. So that inhibits the death signals, I guess, if you just stop. So we call that quiescence, or these are drug-tolerant persister cells. And that's a pretty active area of ongoing study, is what is protecting them, I guess, from dividing, protecting them from needing that oncogenic signaling, which they usually are addicted to.
Yep.
Now,
and I will say, like, I can use my example as a prime example is, after I had surgery recently. We covered it on another podcast, but I was on an ADC at the time, very briefly, TKI, but they removed my tumor and it was still 30% viable, which is exactly what you're talking about, right? So even though I was responding exceptionally well, I was having great scans and a great response, I still had 30% of my cancer that was like, hey.
Yeah. Well, and you weren't progressing.
Right.
So the cancer cells were just hanging out.
Yep.
And there is some nitty-gritty science that has, we have some understanding of that, but it's not to the point where we can target it or prevent it yet. So for that element, I'll say, I'm not sure yet. Now, once we get to how do cancers then progress, there's usually a couple of ways I think about. Now, we mentioned that the protein that's mutated, it's a three-dimensional structure. It has grooves, it has pockets. And in one of those pockets, usually, where the term kinase came from is because that's how it's interacting with other things, this kinase domain or this kinase pocket, the signaling pocket, that's usually where the mutation is. Not always, but usually. And we have a drug that goes in there and blocks that from interacting with stuff. Sometimes you can get another mutation. And that other mutation, it just happened by random chance. The cell is not intentionally thinking about this. It's not designing resistance mutations, but by the random chance of there being millions of cells, one of them had another mutation. And that mutation was functionally important because the drug that you had bound right there. And because that mutation happened, it changed something about that pocket and the drug can't bind anymore. So what we call that is on-target resistance. The target is the protein, the oncogenic protein, the kinase protein. So we call that on-target, meaning the target itself developed resistance. And that can be very, very fortunate for us because if one drug is resistant there, sometimes another drug will work just fine. You can use something else. You can use a different tyrosine kinase inhibitor. This is a strategy we use a lot in examples EGFR, ALK-driven tumors, where if you have an on-target mutation, a lot of times you can switch to a different drug, and the different drug will bind somewhere slightly different in that pocket, and it won't be affected. So that can be a very, very effective strategy if on-target resistance occurs. Did I explain that element okay, Samantha?
Yes, you did. I'm tracking.
No, we're going through complicated stuff, so I want to make sure I don't breeze over things I should be elaborating on.
So why then, I'd love you to dig a little bit deeper into this. So there are some mutations where drugs can work a very long time, and other mutations not so much, although there are obviously the outliers, for sure. There always will be, right? Is that because the mutation is less aggressive, or is that because the drug is better?
It's a tough question. I think it's probably both. And I'm going to say probably because it's really hard to know anything with certainty here. But I will say that in my, I run a laboratory and we grow oncogene addicted cell lines. So some cell lines will grow much slower than others. Some oncogenes are not as strong as other oncogenes. So I think that's true. I think at least in some of our examples with patients and some of our examples in the lab, it does seem that some of the oncogenes have less of the signaling. But also some develop mutation. How do I phrase this? Some of the tyrosine kinase inhibitors are just better. So some of them bind specifically really strongly in a pocket, and some of them bind more weakly to that protein. The ones that bind really strong do really well. The ones that are in areas of that pocket where the cell can't really develop another mutation there, or it'll just stop working. So there's some additional science that can help make those better. But yeah, I think it's a combination of things as to why some oncogene-driven tumors have better responses than others. Let me give you one of the first examples to speak to the differences here. So the example that made me really eager to get into this work back when I was a wee boy was chronic myeloid leukemia. So this is an example of a cancer that's driven by not a single point mutation where one bit of protein is swapped out for another. It's actually a fusion where two proteins get fused together, essentially. And the proteins are BCR and ABL. They stand for things. And those get fused together, and the ABL part is the kinase. So that's just always signaling now. You have ABL kinase. And this guy, who his name is Dr. Brian Drucker, he said, hey, these cells have this new fusion, this new protein that's not supposed to be there. Can we knock that out? And will that cure them? And to my knowledge, he was really the first person to see that there's something scientifically different about these cells. Can we target that thing and that thing only, and it'd be good enough? And by and large, it was. So he came out with Abl kinase inhibitors, or he worked with drug companies who were making them. And the first drug was called imatinib or Gleevec. And now individuals with chronic myeloid leukemia from Gleevec and subsequent better iterations of that can live nearly normal lifespans.
Wow.
I mean, we're talking 20, 30, 40, 50 years. And why that's inherently different from many of the other ones we see, I'm not entirely sure. He may know. And actually, hearing that story was what, in undergrad, was what sparked me to go into this. And then I worked in his lab when I was in med school. So he was at Oregon Health Science University. So I went over there for a summer to work in his lab. And he has he had a book that he wrote, and I had him like sign it and everything. He was definitely my science idol for, forever. He's made massive contributions to the field through that work. But that was the first demonstration that you could do this, you could accomplish that. And that worked really, really well. And then that sparked a huge landscape of what can we find that's mutated and make inhibitors too. And by and large, nothing's worked as well as that first thing. So I think a certain element of it was luck. Yeah. And he'll tell you that. Anyone who works in science and molecular biology will tell you that any success that you have is there's a certain degree of luck. Many, many great ideas fail just by chance. So I can't, very long-winded way to say I'm not really sure, but some work insanely well, and some work poorly. Yeah, it's a combination of the protein itself and the drug we're using, probably. Yeah, there's also this element of how much do other areas in the body need that protein. So let me give you another example here. Let's use EGFR. That's a better one.
That is a better one. I know, because I know where you're going, so it's a better one.
Yeah. So the epidermal growth factor receptor, EGFR, commonly drives lung cancers, at least. It can drive brain tumors. It can drive other things with mutations, but lung cancer is mostly where it's been studied. And the EGFR protein, it's an oncogene. It's a kinase. It can get mutated, and it can tell the cell to keep dividing. And that's how you get these EGFR-driven lung cancers. EGFR is a normal protein that other parts of the body require. So it's on your skin, for example. So people who take EGFR inhibitors, these tyrosine kinase inhibitors, their tumors respond for a long time, in the order of years, with the most up-to-date one. Not for everyone, but for many people. But a lot of times they do have these toxicities because they can get skin rashes, they can get brittle nails, they get hair issues. Because EGFR, you need it in different parts of your body. So we're always struggling with this balance of how can we target the tumor without targeting the other parts of the body. And that was the whole idea of these things, right? Like, that was the difference between these and chemotherapy. The reality is this is just not always true. Sometimes the drug knocks out that specific protein, but that specific protein is needed elsewhere. One of the better advancements that's been coming along is we're getting more and more medicines that are more specific to the mutation, which is pretty hard to do. Yeah, so that's an advancement of the field where we can say, oh, it'll only bind if this thing's mutated. It'll not bind to the normal ones, or at least not as well. That's a hard chemical ask to make a compound that does that. But if you want to know why cancer development takes so long, or cancer therapeutic development takes so long, and why it costs these billions of dollars, it's because they're trying to make tons and tons and tons of compounds that work better, that are more selective for mutated proteins, and to not attack the other healthy stuff in your body. Now, earlier, you gave an example where some people can get a tyrosine kinase inhibitor and it works really well, and they don't, maybe you have fewer side effects. There are examples of kinases that are upregulated in cancer, or they're mutated in cancer, and they're not really there in the normal adult human. Some of these are things that happen as we're developing, like in utero, in the womb, and you have to make, your body has to make different types of tissues, and it's changing But once you develop those tissues, this protein isn't used anymore. So you can hammer that thing. Your doses can be super high, and that is an advantage if the protein isn't usually around in the adult human. But it's not super common.
Mm-hmm.
And a lot of these kinases look pretty similar to each other. So it's pretty hard to make a compound that attacks one and doesn't attack any other one.
Right.
That's very, very, very hard to do.
So the $1 million question for me, before we go into maybe some side effects and stuff, is, along the same lines of me saying, why do some work for a long time and others don't? Why do some people respond so well to these and get 5+ years, and then other people might get like 6 months? I guess that's a $1 million question that we don't know, right? Need to do more research, but
I was just thinking that I answered part of your question earlier about progression and what causes resistance.
Yep.
And that's, so one of those things is this on-target resistance where your protein can get additional mutations that prevent the drug from binding. So that's one of the ways.
But why does it happen so quickly in some people versus others?
Oh, I don't know.
Okay.
I don't know. This is a kind of ongoing question is, are there clones of that cell that were there in the beginning? Like your cancer keeps growing, it keeps growing. Every time it divides, there's a chance for a random mutation. And cancers are dividing so quickly that they don't do as good a job repairing their DNA. So they're more prone to develop mutations. So was that mutation there just by chance, just cause? And then you give your tyrosine kinase inhibitor, and that's just what survives. That's kind of my prevailing theory. Or, is there a certain adaptation that the cell has under the TKI pressure? You give the TKI, some stuff lives, and then it's just mutating to try to figure out a way around it. That's possible too, I suppose.
Yeah.
I don't have the exact answer, and I don't know why it happened so quickly, but for some people, it does happen very quickly. And is it happening because the mutation was just already there? That's part of my thinking. But there are a couple other ways that the tumors can not really care about the tyrosine kinase inhibitor being there, even though it was addicted to it. And I think of them in two buckets. And again, this is just kind of what we know. So one is that on-target resistance. So you're looking for another mutation in the gene you had. So for example, HER2, we put you on a HER2 tyrosine kinase inhibitor, or HER2 TKI, and then you have another mutation in HER2. And I would say, okay, this is an on-target resistance mutation. Do we have another TKI that might work here? It's worth looking into. The other buckets are, it's still dependent on a kinase of sorts. I mentioned there's a bunch of them. The oncogene that's in a given tumor is one option, but it can upregulate other ones, and they don't have to be mutated. So that's a really important concept, is maybe the tumor started from a mutation, but the mutation led to more oncogene signaling from that thing that can't be turned off, but it can get to more oncogene signaling in other ways too. It can upregulate other types of these proteins. For example, in lung cancer, we see upregulation of a protein called MET. It's not a mutation. It just makes a bunch of it. It's trying to survive you cutting off that addiction. And a lot of these tumor cells will upregulate a lot of different oncogenes. And they'll just send these signals out, help me survive, give me input from other kinases, and it'll end up being enough to survive. And then usually, or maybe not usually, but I'll say enough times that we can study it, it will become dependent on one of those. And then we could potentially target that. So that is a well trialed strategy where if someone, for example, is given a HER2 inhibitor, the cancer cell is starved from HER2. It can't get any of that because the drug works. It'll upregulate stuff. Let's say it upregulates this protein MET. And all of a sudden, we can stain for that. Looking at the cell, we can see how much of this protein does it have. And if it's on fire, if it has a ton of it, then we'll say, okay, let's go after that. Let's target that either with another tyrosine kinase inhibitor or target it another way.
So this is a side note real quick, but I think it's important because it's come up a lot in some of my groups recently is that MET thing that you're talking about. When people progress, they're like, well, have you tested for MET? Is that a different test, or is it a same biomarker test that you get done?
Good question. So I mentioned that a lot of times these oncogenes, these kinases, are signaling because there's a mutation, but it's not the only way they do that. So what you're usually getting from a blood test is what are the changes in DNA? And DNA, as I mentioned, is that template. So the template can be wrong and it'll just make protein that signals too much, or the template can be totally right and your cell is just making too many of those proteins. It's a conveyor belt, and if it's supposed to make one MET every whatever minutes, now it's making 50. The DNA may not show that. It may show that if instead of, usually have 2 strands of DNA, one from your mom, one from your dad, sometimes it can get amplified where you actually have many, many more. The blood test can usually detect that, not always, but usually, or the tissue biopsy, whatever's looking at DNA can usually detect that.
Okay.
But that's not always how it happens. Sometimes it just makes more, and I can't tell you why.
Okay.
And then you have to look at the protein, because a lot of times you're inferring from the DNA, if the DNA is amplified, like you have more copies of that gene than you're supposed to, we're assuming it's going to make more protein. And a lot of times that's a fair assumption. But, the proof's in the pudding. You got to look at what the protein is doing. So I'm an advocate for a lot of my patients. I do look for MET. I do look for the alternative pathways that could be activated if there's something you can do about it. Because there are a lot of kinases out there, but we don't have therapies to all of them. Actually, we don't have therapies to most of them. So right now, the field we're coming along, but we're still pretty young in that. But yeah, to your point, it's not all in the DNA. Sometimes you can look and see what's happening at the cell surface, and can we target that?
And is that through like a tissue biopsy, or how do you find that out if you can't get it from the blood? Okay.
Yeah, protein, you have to look at the actual tumor cell itself.
Okay, gotcha.
Like visibly look at it.
Okay.
DNA is, it's biochemical. You're like, it's reactions. But when you're looking at protein level, what they're doing is they actually have antibodies that are specific for that protein, and they don't target anything else. And then you add the antibody to the tumor and you look at it under the microscope and you say, is that antibody there or is it not?
Oh, interesting.
It's like not terribly complicated.
Okay.
And then we have people, pathologists, who are rating how much is there. Is it a little bit? Is it a medium bit? Or is it a lot of it? And that's literally the test. It's 1, 2, 3, little, medium, lot. And if there's a lot of it, then I'm like, okay, well, that's let's give it a shot, So that is a well-known way that tumors can evade tyrosine kinase inhibitors. There's a third way. The first 2 ways I just mentioned still depend on some form of kinase signaling, some form of oncogene signaling. The third way is what we call lineage change. And that's complicated. So a lot of these oncogenes we're talking about happen in tumor cells that pretty much resemble what they started as. If it started as a bit of lung, it looks like that. It's just more of it. But things can devolve. They can change. Tumors can do this. And eventually they can become no longer dependent on that oncogene signaling. It's almost like when I was mentioning before, if someone's exposed to a lot of carcinogen, their tumor just gets it gets dysregulated. There's too many mutations. It can't handle it. There's too much signaling, not too much signaling to divide, not enough signaling to not divide. And they can eventually turn into that. And we do not have great therapies when that happens. We have some, but we don't, the tyrosine kinase inhibitor field stops really working there. There's science coming out that may better target that, but we've been pretty unsuccessful once it stops caring about oncogene signaling. And then we go back to things that just work on dividing cells like chemotherapy.
Right.
So that kind of gets to what happens when we no longer have these tyrosine kinase inhibitors left, or if someone's had on-target mutations, meaning the protein has a mutation in it, the protein you're targeting, you try a different TKI, maybe that works, it can happen again. Eventually they're going to stop working because we run out of ones that work. Same's true with if it starts being dependent on a different oncogene, eventually you're going to run out of things to use. Or if it undergoes this lineage change, the tyrosine kinase inhibitors, the TKIs, aren't going to work. And then you are left, the most commonly used standard of care after that is chemotherapy. And again, these are big generalities because we're talking about a bunch of different tumor types. There are many of these oncogenes. And they're not only, you and I are in the lung field, but they're not only in lung. Once the TKIs stop working, you're really looking mostly at chemotherapy-based strategies. So that can be chemo directly, like the first line of therapy you had was chemo.
Mm-hmm.
And then after that, we can do things that deliver chemo, like antibody drug conjugates. That was the next thing you got. And it wasn't a coincidence you got them, before, because your TKI wasn't approved yet.
Correct. Yes.
So the things we usually give are what you got when we don't have TKIs that work anymore.
Right.
So you had chemotherapy, you had an antibody drug conjugate, which is an antibody conjugated essentially to chemo. And then as long as the cell is expressing its target, you can attack it on the tumor cell. And then, we can try things like immunotherapy, which hopefully by this point, the cancer cell is mutated. It's different enough that the immune system might better recognize it. So I think it's possible that immunotherapy works better now than it would have when initial diagnosis happened. I don't have great data to support that, but theoretically, I think it makes sense. And then there's, the clinical trials, which I'm always an advocate for clinical trials pretty much as early as you can get them. If there's one available to you that makes sense. But that ends up largely being the landscape once unavoidable resistance to TKI has occurred, or resistance you can't mitigate.
I have a question that's potentially going to put you on the spot, so I apologize in advance. There are some people who, they'll do a stint of a TKI. They'll maybe then, because they've progressed, go on chemo or some other ADC. And while I know this is not the standard of care, sometimes a re-put on the TKI. Again, I know that's not standard of care, but it's more of a let's try again, why not kind of thing. And I think in some cases it seems to somewhat control things again, maybe not for as long, but for a little while. And so I wonder if you could speak to that a little bit about those rare circumstances where it might work again. Why suddenly would it?
Yeah. So it's a complicated topic for sure. And I'll phrase this, I suppose, in a few different ways. So one way is that you have a large volume of cells that are surviving, I suppose, the TKI, but many of them are not dividing. And the TKI is probably responsible for that. If you take off the TKI, you already knew about the area that was progressing, because that's why you're changing treatment, but you're not really thinking about all the areas that aren't. So in my mind, the TKI is actually doing a good job at those areas.
Right.
It's just, maybe 2 or 3 or, wherever areas that the TKI is not working on. So in my mind, if you have something that still works where the TKI was working, then it makes sense to totally switch. But we do have some data that doing chemotherapy and then continuing a TKI or something like that can be helpful in certain circumstances. And again, I want to reiterate that every circumstance is so unique that this is a situation where, talk to your doctor about it. Absolutely. But the mechanisms of resistance are different. The mechanisms of resistance to chemotherapy are very different from the mechanisms of resistance to a TKI. So if you think about like a Venn diagram where on one circle you have the things that are resistant to a TKI on a different circle, or the things that are even resistant to chemotherapy, if you have them both at a given time, you have to have the small minority of cells that are resistant to both. So it does make sense that if there's progression happening, it may make sense to use them both. The question isn't necessarily, is it conceptually beneficial? It's, is the extra toxicity of the chemo worth the benefit?
Right.
And that's where the trials have to answer it. Because there's a lot of stuff that we can do in the lab, and this is something we can do in the lab, and I promise you, you're going to kill more stuff. But whether it's worth it or not in a human biologic context is where the trials need to play in.
Which is a great segue to toxicity discussions.
What was your perception when you first started TKIs about the toxicity compared to chemo?
I was under the assumption I would have some side effects for sure, but it wouldn't like destroy me per se, and it would be a much more manageable it would just be much more manageable, and I'd be able to lead a pretty normal life, which
Has that
was very
been
exciting.
the reality?
Oh, very much so. Yes. I mean, I will say the one thing I was terrified of was diarrhea. I think every TKI that I know out there for lung cancer produces some form of diarrhea potential. And so that was my number one fear, of maybe having like bouts of uncontrollable diarrhea, or those kind of scenarios play in your head. But for me personally, I've been very fortunate where I have not had any side effects. I mean, maybe I'm possibly a bit more tired than I was before, but nothing I can't push through.
Yeah, and I think what you're describing is a lot of people's experience on TKIs, but it really depends. So as a field, it's not as consistent as chemotherapy is, that many people are going to get nausea, and many people are going to get, diarrhea, and many people are going to potentially get neuropathy and things like that. But even that, within chemotherapies, they're different. And we have had different podcast discussions on different types of chemotherapies. Their side effect profiles are really different. TKIs are no different, but I would say as a field, as a class, they're a lot more manageable than the chemos are. I mean, for example, many chemos are limited in the number of times you're going to get it because we know the toxicities are too bad if you stay on it. And that's not usually true with the TKIs. Most of the time, people can be on these really indefinitely. I'll say, for those who are listening and were maybe getting a TKI 10 years ago, you'll say, I beg to differ, because the engineering of these wasn't that good. It wasn't as good as it is now when it was first coming out, because we didn't realize, or I suppose we didn't have enough understanding of how to specifically target the tumor that's mutated. I gave you the example of that EGFR protein, where if you target it, you can get all the skin stuff and all the hair changes, nail changes, diarrhea. And as we've gotten better at designing these, the toxicities have largely gotten better. Let me add, that's if you're only targeting the protein you want to target. Many of these kinases are what I would call dirty. So if you look up the targeted kinase inhibitor that of interest, you can look at its kinase inhibitor profile and see all the things it targets. People study this. So, there's this idea of the kinome, which is not the chyle-ome. It's all of the kinases in the cell, and what is the degree to which your drug binds to different things. So, for example, the first class of targeted kinase inhibitors that came out, and say, I'm going to give an example still on lung because the contrast is strong, crizotinib in ALK-driven tumors. Crizotinib was the first drug that came out for ALK-driven tumors. It was the first targeted kinase inhibitor, but it was really dirty. It attacked a lot of different things. So the side effects were really bad. It was really hard to tolerate that. It was tolerable, like people could do it, but it wasn't that much of an improvement from chemo. And then as we got better in our engineering, now the side effects are significantly lower because they're just inhibiting ALK by and large. And as the science continues to progress, and this is another reason to continue investing in TKIs, we do get better it's better at attacking the actual protein and not attacking other things. That being said, I think the big caveat to tyrosine kinase inhibitors is that most people don't have these types of mutations. So really, along the spectrum of cancers, most people are not going to be on these, at least not right now. So that's a big challenge. Is they're very exciting, and I think they appropriately get a lot of funding for development, but it is a small slice of the cancer pie that's benefiting. So it's tough. A lot of times I would say, as a thoracic oncologist, we're really hoping when we see the tumor profile that we see one of these, because it gives us more tools. If you're gonna get any cancer, you want one that you have the most tools for. So we're hopeful that we see something like that once a tumor's been diagnosed, but many people don't have these. And they are using chemotherapy, and they are using immunotherapy and other strategies, the antibody drug conjugates. But the field is developing. We're learning more and more about what we can use these pills for. So there's hope that people who are not currently eligible for these targeted kinase inhibitors become so at some point in the future.
It's a very exciting time. I keep saying that I timed my cancer very well in terms of having FDA approval on the first HER2 TKI at the time that I needed it. So
yeah, and we've talked about this before. There is an element, I think, of the emotions are complicated around being an individual who can or can't receive these, like your tumor, meaning is susceptible to them or not. I think one thing I've heard from the community is sometimes there's a lot of guilt in people who have targetable mutations that tyrosine kinase inhibitors can attack. And I wouldn't say there's so much as disdain or anything, but there's a tough feeling if you're someone whose tumor doesn't have that. And to what you were saying earlier, do we know what causes these? And we don't know. And a lot of it is just, it's at least the level to which we understand now, we call it bad luck, because whatever thing caused it or whatever prevented your body from being able to repair that damage, the DNA damage that caused the protein, I don't know. I don't know why it happened in one person and not another person, but I do know in a lot of the communities, and even within, I'm sure you could speak to this, even within tumors that have kinase inhibitors, some work better than others. And that's a tough feeling when
there's a lot of envy.
Yeah. And it's not the patient's fault. But it's hard, it's hard to learn a lot more about this field and not have strong emotions because our lives are on the line, people's lives are on the line. So it's tough. So I guess my recommendation to individuals who are receiving these tyrosine kinase inhibitors, or if you're listening because a loved one is potentially going to have one of these, no matter what bucket you're in for an oncogene, there are no guarantees in this field. I know people who have been on oncogene targeting inhibitors, these TKIs, for years and years and years, when it would have been predicted for less than a year. And I know people who would have been predicted to be on this for 5 to 10 years, and they progressed within a year. Nothing about this is fair. The tyrosine kinase inhibitors are not a perfect solution to cancer. They do not cure cancer, but they do, I think, represent a really positive next step from chemotherapy to specifically attack the root cause of a cancer and minimize side effects that the rest of the body would experience from chemo. So I think they're a huge advancement, but they're not yet the cure-all that we hope one day they'll be.
One day.
Well, I think we need to combine things.
Yeah.
And we are. But I think, I'm a big advocate for development of cellular therapies and things like that, which are reprogramming the immune system to attack cancer.
Which is a podcast for another day.
But, we need to be joining combining a lot of different therapeutic modalities together. As much as I like chemotherapy-free options, recent data suggests maybe people with oncogenes can benefit from chemo with their tyrosine kinase inhibitor. So the field keeps evolving. Everything's more complicated than you think it is. But we have a lot of dedicated people who are working to make this better, working to provide hope. And the field is getting better. Things are improving. Samantha, when you were diagnosed, what you're on right now didn't exist.
Mm-hmm. Yeah.
Well, it existed. It wasn't approved.
Yeah. Yeah.
Yeah. Samantha, what last things should we cover before we wrap up here?
I think you've covered everything great. Explained a lot, answered my difficult questions. So you're good.
No, I think whenever we do a podcast, I'm ready to phone in the editor and say, hey, section's here to here. No. I think we covered a lot of stuff. And yeah, hopefully this answered folks' questions about what a TKI is, what it isn't, some general expectations. It's hard to get too specific because there's so many of them. So this is just introducing the field. So when an individual can walk into the oncology office, they have some sense of what these terms are that are being thrown around, and maybe how to feel about it. All right. Well, I think that's as good a place to cut it as any. Thank you everyone for listening, and thanks Samantha for joining today.
Thank you.
Well, everyone, this has been Cracking Cancer. You can subscribe and visit crackingcancer.org for more information. We'll catch you next time.
Thank you for joining us on this episode of Cracking Cancer. If you found this helpful, please subscribe, leave us a review, and visit crackingcancer.org for more information. Every dollar we earn goes directly to furthering the podcast and supporting vital cancer research. Because together, we can crack cancer.